the one dot to the next is catalyzed by some enzyme. And it's catalyzed by an enzyme. So this is metabolism. Instead of reactants moving from one reactant to another, catalyzed by an enzyme or a catalyst. Now, there are two kinds of metabolisms, autotrophs and heterotrophs. Autotrophs create the metabolites, and heterotrophs eat them. So plants are autotrophs, and we are heterotrophs. Now, there are two kinds of autotrophs. There are photoautotrophs, which depend upon light for their source of energy. That is the chemical equation for photosynthesis. Chemoautotrophs depend on inorganic molecules, such as hydrogen, to reduce carbon dioxide. Chemoautotrophs are those that make their food by chemosynthesis. Chemosynthesis is a process by which some organisms, such as certain bacteria, use chemical energy to produce carbohydrates. They are capable of utilizing inorganic compounds such as hydrogen sulfide, sulfur, ammonium, and ferrous iron as reducing agents and synthesize organic compounds from carbon dioxide. Chemoautotrophs are found in hostile habitats such as deep sea vents and where light cannot easily penetrate through. They include the methanogens, halophiles, nitrophiles, thermoacidophiles, sulfur oxidizers, etc. Now, there are two dominant theories right now in the origin of life. The first one is called metabolism first, and this is what I'm talking about, metabolism. The second one is the genetic code first, RNA, the RNA world. Now, I am a deep and honorable member of the metabolism first. So today I'm going to be talking about metabolism. My next talk will be on the genetic code. So I went to, ChatGPT to find out. So you can read it rather than me reading it. All right. For me, the metabolism first idea first started in 1975. This is the paper which I wrote in 1975. An autotrophic origin of metabolism is described, which requires clays, transition metals, disulfide, dithiols, UV, and cyanide. A general scheme is proposed involving the fixation of CO2 and nitrogen for the evolution of intermediary metabolism based on the evolution of a complex system from a simple one. The basic conclusion is that metabolism could have evolved from a simple environment rather than from a complex environment. Autotrophy means no soup, because the biggest theory in the non-metabolism first is that there was a soup, and the monomers were there for you, and you then built from backwards from the monomers. All right, the citric acid cycle. This is now from the paper. If we look at intermediate metabolism, in particular anabolism, that means building up from CO2, nitrogen, and all the rest, we are immediately struck by the central role played by the citric acid cycle, that cycle that you see there. And if we apply the method of Bernal, which I will tell you about in a minute, it can be conjectured that the citric acid cycle came first and was followed by the amino acids, lipids, nucleotides, and carbohydrates. There's a little something which I'm going to talk to you about next, is that from the citric acid cycle, there is the number of steps that amino acids are made. So from alpha ketoglutarate up there's one step which takes you to glutamate, another step which takes you to glutamate semialdehyde, which then cyclizes into proline. And there's a split there. This thing becomes ornithine, citrulline, and arginine. So from the citric acid cycle, you can look at how, here's the citric acid. Let's look at the citric acid cycle just to get you acquainted with it. There is the dicarboxylic acids, and there are the tricarboxylic acids. The dicarboxylic acids go from oxaloacetate through malate, through fumarate to succinate. Up there is the aconitase, isocitrate, and coming down to alpha ketoglutarate, which is turned into a dicarboxylic acid at that point. There are not that many tricarboxylic acids, but it's mostly dicarboxylic acids.
Is this the same as the?
That's the same thing that I showed you in the beginning.
Let me finish. As the Krebs cycle?
Yeah, it's the same term.
Okay, well, I didn't know that.
All right, now, the interesting thing was that when I was at Berkeley, there was a big controversy between the citric acid cycle, not the citric acid cycle, but between Arnon and Calvin. Calvin had this Calvin cycle based on sugars. And Arnon discovered this reverse citric acid cycle, and Arnon and Buchanan suffered it. And it was very controversial. It was a huge controversy at the time. So therefore, I never started with the reverse citric acid cycle. All right, the metabolic metric. This is very important for the next talk. From a more detailed examination of metabolism, some amino acids, this is coming from the paper. Amino acids require fewer steps in their biosynthesis from the citric acid cycle. Glutamic acid, glutamine, proline, and arginine are closely linked metabolically to ketorate. And the rest of it just talks about some of the other ones. The metabolic metric, and this is all you have to remember, is the number of metabolic steps it takes to synthesize an amino acid from the citric acid cycle. Now, the onion heuristic, the Bernal thing, this is now going to be used over and over again, not only today, but in my next talk. This is that evolutionary systems start simple and grow like an onion, and they build on top of each other. And therefore, you can peel them back. That means that the history of metabolism, that big chart that you saw, started off simple, evolved in an onion-like fashion, and therefore you can reconstruct its history. This means that you can go much further back in time to the origin of life.
Can I just ask a question?
Sure.
When you get to the core of an onion, the core of an onion is not the same as the seed that gave rise to it. So
Exactly.
From the core to the seed.
No, there has to be some system that is remembering and is evolving. These are evolved structures, not steady state structures or self-organizing structures.
Yeah, but there's but I'm just saying, like this memory seems to have a limit.
Well, anyways, can we go on? It's going to evolve because it's going to evolve into the genetic code. It doesn't start with the genetic code. It starts with self-replicating clays, which are the memory system. Okay, the idea it's a very nice quote. Life has developed its process gradually, never rejecting what it built, but building over what has already taken place. As a result, the cell resembles an archaeological excavation with the successive strata on top of one another. The oldest one is the deepest. The older a process, the more basic a role it plays, and the stronger it will be anchored. The newest processes dispense with most easily. The idea was expanded by Bernal into the conceptual tool for studying evolutionary processes. The method describes how a complex system evolves from a simpler system. The pattern is one of stages of increasing inner complexity following one another in order of time. Each one included in itself, structure and processes evolve at the lower levels. The system thus grows more complex like an onion, and it must be possible to strip back the later layers of the onion to expose the simpler one. Now, this was in 1975. All that stuff was basically in 1975. You can look at the paper. Now, in 1992, photosynthesis, conjectures and reveries. Okay, and I'm now looking back to my former paper. In a previous paper, a autotrophic origin of metabolism was described, which involved replicating clays, transition state metals, and UV in the fixation of carbon dioxide and nitrogen. Hartman said 1975. The origin and evolution of photosynthesis can be related to the evolution of those self-replicating and evolving iron-rich clays. 4 distinct phases can be discerned in the evolution of photosynthesis. The 4 phases may be labeled the iron world, the sulfur world, the nitrogen world, and the phosphorus world. The system evolved as an onion. Each world included the previous world. One can thus strip back the phosphate world to uncover the nitrogen world, and then back to the sulfur world, and then back to the iron. That was what was proposed in 1992. Now, what are these 4 worlds, basically? The first phase was dominated by the photoreduction of carbon dioxide by ferrous iron and zinc sulfide to form glyoxylate and pyruvate, which are the precursors of the reverse citric acid cycle. The second phase was the entry of sulfur into the evolving clay system, which led to the formation of these ferredoxins, Fe2S2 and Fe4S4 cores, and acetyl thioesters. A reductive dicarboxylic acid cycle, which involved acetyl thioesters conducting carbon dioxide to form pyruvate, which condensed with carbon dioxide to form oxaloacetate, which in turn is reduced to form malate. The malate then splits into acetyl thioester and glyoxylate. This was something that Ivanovsky did. This cycle was later expanded to form the reductive citric acid cycle. A variant of the reductive citric acid cycle would be driven by reduced ferredoxin analogs and thioesters. Okay, any questions about that?
So this
Yeah,
before the ATP cycle.
This before, no, this is the citric acid cycle. This is reductive citric acid cycle.
Right, but at the moment, this reductive TCA cycle is driven by ATP.
No, it's driven by thioesters and ferredoxin.
Modern organisms rely.
Oh, yeah, modern organ, but that's not what happened back there. We're in the sulfur world right now.
I'm stuck on number 1. I thought ferrous iron can reduce carbon dioxide
Yeah, sure.
by itself without.
Yeah, but
Photon.
reducing carbon dioxide is not enough. You want to essentially build it into.
Okay, my question is, why do you call it photoreduction? You just put them together and it'll.
No, photoreduction is you need reducing agents that are driven by light. That does
Well, what is
does the reduction.
serpentinization?
What?
What is serpentinization?
Oh, serpentinization has nothing to do with this.
Okay.
There's nothing to do with serpentinization at all. This has to do with iron-rich clays. Okay,
The iron naively, I don't think is powerful enough to reduce CO2 just by itself. So what you're saying is that you get something like sulfate.
well, I need a zinc sulfide. which is a more powerful agent.
Right.
But I said zinc sulfide. But I called it the iron world because you'll see later when I come to,
I mean, one question about that is that, is it possible that if you just change like a ligand structure on iron itself, you can change the reducing potential? Like, it doesn't have to be sulfur, but some organics. Like, so is it?
yeah, sure.
Reducing-
Right now, I'm doing what I thought in the early days of the. Okay, hold a second. Third is the ability to fix nitrogen. Now that just came up, by the way. It turns out that pyrite in the UV in water will fix nitrogen. That was been a problem. With the fixation of nitrogen into ammonia, the biosynthesis of the amino acids from the citric acid cycle becomes possible. The first polymerization of thioesters of amino acids would result in the first peptides. These peptides were possibly made up of structural hydrophilic amino acids and form the first peptide membranes. That'll come in next week when I talk about it. In the final phase, phosphate entered the evolving system. This resulted in fixation of carbon dioxide by the reductive pentose phosphate cycle, Calvin cycle. The formation of nucleotides now become possible, which led to the fluid mosaic membrane. That's a combination of peptides and lipids. Finally, the synthesis of nucleotides and polymerization of nucleotides in polynucleotides join the peptides of the thioester world to result the origin of the genetic code. That's next week. Where did this take place? On the primitive Earth. Did life begin? Where on the primitive begin? The origin of life and photosynthesis would take place in a hot spring on the surface of the early Earth. The water in the hot spring would be rich in ferrous iron and other transition metals, molybdenum, copper, zinc. The water would also contain magnesium ions, aluminum ions, silicate ions, and gases such as CO2, nitrogen, and H2S. After the sulfides had precipitated out, the excess ferrous magnesium silicate would form the iron-rich clays. These clays would fix carbon dioxide into oxalic acid and other organic acids, which can catalyze clay formation itself. These replicating iron-rich clays would evolve into the sulfide region of the primordial hot spring, acquiring nitrogen, fixation in the process. Finally, phosphate is incorporated into the evolution system. The four phases now, I'm going to remind you, will be labeled the iron world, the sulfur world, the nitrogen world, and the phosphate world. The system evolved as an onion, each world including the previous world. That's just to remind you, one can thus strip back the phosphate world back to the nitrogen world, sulfur world. The nitrogen and sulfur worlds are related to each other very closely. Now, I just want to remind you historically, this idea that photosynthesis was the key to the origin of life was done by a very famous biochemist, Sam Granick. And what he said, and this is the article, it's a nice article which sort of sums up his stuff. Living matter is an organized system which requires a continual flux of energy for its survival. The iron oxide sulfide photosynthetic unit proposed by Granick 35 years ago was meant to supply this energy. That's a long time ago. This was written in 1992. The Granick hypothesis that biosynthetic pathways recapitulate their evolution. So therefore, biosynthesis recapitulates biopoiesis. This is sort of a taking off on ontogeny, recapitulating biology. So photosynthesis in the origin of life. The origin of evolution is considered to be the key to the origin of life. This eliminates the need for a soup, and the synthesis of bioenergetics comes from the fixation of carbon dioxide and nitrogen. No soup, then no RNA world or protein world. end of story. That means there's only one thing now, metabolism. The RNA world doesn't exist. The origin of life is considered to have occurred in hot springs, blah, blah, blah. If biosynthesis recapitulates biopoiesis, then the synthesis of amino acids preceded the synthesis of purine and pyrimidine bases. The polymerization of the amino acid thioesters into polypeptides preceded the directed polymerization of amino acid by polynucleotides. Thus, the origin and evolution of the genetic code is a late development and records the takeover of clay by RNA. That is talking about how this thing expanded its evolutionary world. And now, where do we ever get these iron-rich clays? Well, we know from the stuff on space, the meteorites are all filled with iron-rich clays. And it turns out on Earth, this is this guy, Alan Munier, who works with the people here. Considering the high catalytic potentials of clays, and particularly those of the iron-rich ones, electron exchangers, it is probable that large parts of the surface of the young Earth participated in the synthesis of prebiotic molecules during the Hadean to early Archean period through innumerable clay-rich settings in the massive parts and the altered surface of comatite and basaltic lavas. Iron magnesium clays, this is the clays that I've been talking about, were formed inside chemical microsystems through sea weathering or hydrothermal alteration, and for the most part through post-magmatic processes. This leads us, this is what he says at the end of this paper, to suggest that iron magnesium clays should be preferred to aluminum-rich ones, montmorillonites, to conduct experiments for the synthesis and polymerization of prebiotic molecules. That should be opened up now, because the iron magnesium clays have been made here in the hydrothermal things in order to test the reflectance spectroscopy of them on Mars. And they could be tried photochemically. So this is a nice diagram from my friend Gutzmann. Don't take this seriously, but it's sort of trying to give you a picture of what would go on in a hydrothermal system at the surface of the Earth. There is the sun, there is the photochemistry, there are all these ions that are floating around. These are the cycles. He has the reverse citric acid cycle done because of zinc sulfide. And then there's the cycles here with iron and manganese. So it's a nice picture of sort of, but don't take it too seriously. I mean, it's something like this. Now, this opens it up to possible experimentation, and I'll come to that later. Okay. So this is, again, I'm trying to now get this down to you, that this thing probably took place in a hot spring on the surface of the Earth, and it's evolved up metabolism in that hot spring. That means this kind of system can be open to experimentation. You here at Caltech have been working on a system that I'll talk to you in a minute by Russell. Russell has worked here for about, I don't know, 20 years? How many years?
10
Huh?
years.
He's worked here for 10 years. This place is well known. He's written numerous papers on the subject on chemo autotrophy, down at the bottom of the ocean, down where all these poor microbes live. And he has this hydrothermal solution coming in, and then there are these bubbles formed in rocks. And the whole thing is now driven by the proton gradient, not by light, proton gradient. Very poor choice.
You need oxygen in the water.
Pardon?
You need oxygen in the water.
No, this is way before oxygen. The only thing he has in what, the only oxidant he has is carbon dioxide.
Nitrate,
Eh?
nitrate. He doesn't believe.
There is no nitrate if there's no
So
oxygen.
his theory assumes that that is going to be the electron acceptor, not carbon dioxide. It's Nick Lane that thinks it's carbon.
Well, he doesn't have anything. Nitrate comes in with oxygen. You can't, yes.
Mike showed ways that you can get that.
Well, anyways, that's.
Mike won. Mike won. He
Okay.
showed Hartman and-
Anyways, let's discuss this. Now, he did something interesting. This was now the origin of cells, hypothesis for the evolutionary transition from abiotic geochemistry to chemoautotrophic prokaryotes and from prokaryotes to nucleic acid. He wrote this paper, I think this is the proposal, that would be some years ago, with Martin and Russell. I have been in controversy with Martin from many, many years ago on the origin of the eukaryotic cell. And I've been in controversy with Michael J. Russell because of his. So he proposed that life evolved in structure iron monosulfide precipitates in a seepage site hydrothermal mound at a redox pH and temperature gradient between sulfide-rich hydrothermal fluids and iron-containing waters of the Hadean Ocean. The naturally arising three-dimensional compartmentalization observed within fossilized seepage site metal sulfide precipitates indicate that these inorganic components were the precursors of cell walls and membranes found in cells today. A number of interesting things can be said of this. I want to now talk to you about a book that just came out related to this chemoautotrophic hypothesis. And this is, I really recommend this book. It's a beautifully written book. And it's all based on the reverse citric acid cycle. And it's all based on proton gradient. So it's a great book, but it's wrong, fundamentally wrong. Anyways, but that's enough. So the book is brilliant, especially when it comes to cancer. It turns out on cancer, we go back to a reverse citric acid cycle. In the mitochondria, instead of burning acetyl-CoA, they make acetyl-CoA with 2 CO2 things. In the case to base chemoautotrophy on the pH gradient, the attempt to get free energy from a pH gradient does not compute. There are 2 major rabbit holes in the chemoautotrophic scenario. One, a lot of reduced molecules such as hydrogen, weak oxidants such as CO2. The reduced molecules such as acetate and formates self-organize into reverse citric acid cycle. They just do that. Question, why go to the bottom ocean when photochemistry gives an abundance of free energy and therefore drives our biosphere? It was a poor choice for a hydrothermal system. What is more, they consider clay, the serpentine, because its formation gives rise to hydrogen, which is just a reduction. They're already in a highly reduced condition. Thus, clays are not invoked for evolution of metabolism, but instead for the production of a redundant hydrogen. Self-organization is good for crystallization, but not for evolution, which is the increase of complexity in time, according to the onion. So that's my statement on chemoautotrophy. Now, I have made a prediction that there are these 4 levels, 4 times that has occurred in metabolism. Now, this is metabolism of the keg. You can see here the citric acid cycle, but it's really hard to read. This is a version which has been sort of edited, which has to do with the various locations. There's the citric acid cycle at the center, And there are various locations where this things off the citric acid cycle go. Let us test the stages of the four, given we have this keg system. And we know about the phases again. I don't want to go through it again, but just remind you what the four phases are. Iron, zinc, basically, sulfur, nitrogen, and then phosphate. Okay, so we test computationally. This is my buddy. I have to give a shout out to Dr. Goldford. He is the master of this paper. And so remnants of an ancient metabolism without phosphate. So we're stripping out that metabolic system without phosphate. We test computationally the plausibility of an ancient metabolism without phosphate. A phosphate-independent network exists within the biosphere level of metabolism. This network displays hallmarks of prebiotic chemistry, iron, sulfur cofactors. This could represent a metabolic fossil of early thioester-driven biochemistry. Correct? I'm not overstating anything. This network is predicted to support the biosynthesis of a broad category of key biomolecules. Its enrichment for enzymes utilizing iron-sulfur clusters and the fact that thermodynamic bottlenecks are more readily overcome by thioester rather than phosphate couplings suggests that this network may constitute a metabolic fossil of an early phosphate-free catalytic biochemistry. Our results corroborate and expand previous proposals that a putative thioester-based metabolism could have predated the incorporation of phosphate and RNA-based systems. This is the quote from the paper. Now, we are going to show you what happens when you remove phosphate from that metabolic chart. And it made Cell. So it made the cell, the cover of Cell. This is what comes out of the system. Now, there's one other thing that's being done here. There's a network in this system already. But you start off with a seed. Here is the seed, and here is what you put into that seed. And you work out these reactions going through the network, seeing what do you get in this network. This is presumably supposed to be the primitive network, okay? Correct? And so this is what he got. Now, note here a couple of interesting things. I mean, one is pyruvate right here. Glyoxylate right here. This is you can see these big centers. And then there's all the amino acids, and all this other stuff there. Now you can put this on the keg and see what the TCA cycle comes from and what it's doing. It's energy, it's a metabolism, uncommon amino acid, glyoxylate. There's some lipid metabolism, polyketide metabolism. So it tells you not only that it's spreading through the keg, it's telling you what it's spreading to, okay? What amino acids and things like that, correct?
The gene cell piece of this figure, but you don't have to use that.
The properties of core phosphate-free suggest that a thioester-based metabolism may have started from a few simple geochemically abundant molecules and expanded to a surprisingly rich, diverse biochemistry, potentially network-level fossil biosphere. Okay, so basically what I said, you strip back, you're suddenly proposed with a metabolic fossil that is thioester, and that is very good. And then he went on, and this is more complicated. Environmentally, boundary conditions for the origin of life converge to an organosulfur metabolism. Surprisingly, environmental sources of fixed nitrogen and low potential electron donors are not necessary for the earliest phases of biochemical. So now we're going to strip back from nitrogen. We strip back from phosphate, we're now stripping back from nitrogen. We now constrain the network by temperature. He does a whole bunch of interesting stuff. Our analysis of possible trajectories indicate that a subset of boundary conditions converges to an organosulfur-based proto-metabolic network fueled by thioester and redox-driven variant of the reductive carboxylic acid. That is capable of producing lipids and keto acids. And here's what he got. And this is the seeds. This is the thing that he's got, and it's all done but with CO2 is coming in here. And you have basically the reverse citric acid cycle. We have gotten back to what is, according to Nick Lane, the very heart of the origin of metabolism and of life. Now, I'm going to say a couple words about the iron world and the zinc sulfur world and the pyruvate glyoxylate, and the rest is free of transition state metals. I'm going to go through this very, very, very quickly. Metals provoke consequences of the reverse citric acid cycle, and this is the group by Moran and Kamila Muchowska. And basically, they claimed that they could get 5 of the reactions of the system. Then they went on, and this is more recently, Muchowska explored the influence of transition metals as potential catalysts in the contrast use of oxidants. In the presence of ferrous iron and under an inert atmosphere, pyruvate and glyoxylate produced a reaction network that spans most of the intermediates and reactions of the Krebs cycle and glyoxylate cycle. Moreover, the intermediates built up and break down to carbon dioxide in a regime that resembles the biological anabolism and catabolism. It was found that oxidative decarboxylation reactions that give malate, fumarate, and succinate were more efficient in the presence of ferric iron. Upon addition of hydroxylamine and metallic iron, the chemical network could extend to 4 biological amino acids: glycine, alanine, aspartate, and glutamate. And that'll become interesting in the next segment. This is controversial. So now that my friend Gutzman has been working with zinc sulfide. And what he got was essentially going from oxalate, which is easy, to glyoxylate, which is hard, to lactate, and then to pyruvate. This, he already sets up the pyruvate glyoxylate system that seems to be key to starting the reverse citric acid cycle. And now he did something else. He showed that he could go from lactate pyruvate to alpha ketoglutarate by fixing carbon dioxide. It's an interesting chemical system. And now I'm going to end up, I hope, with a plausible metal-free ancestral entirely of alpha ketoglutarate acids. This is no transition metals at all, no zinc, no iron. And this is, I'm not going to go through this thing, but he gets the whole reverse citric acid cycle, starting off with pyruvate and glutamate. Now, I'm going to end up talking to you now about something that has been observed by a guy who's been studying metabolism today. It's called phosphorylation and acylation transfer reactions clues to a dual origin of metabolism. And this is the dual order of metabolism. This is based on the phosphate world, and this is based on the iron sulfur thioester world. And what you end up with is here, these are the preliminary monomers, end up with polysaccharides, polyphosphates, glycolysis, glycogenesis, pentose phosphate pathway, that's the Calvin cycle, and polyribosomal nucleotides. It's a sugar system. This thing starts off with carbon assimilation via thioesters. That's the reverse citric acid cycle. Acyl transfers. And then there's something called polyhydroxy alanates. I'm not going to go into those. Fatty acids, peptides, and citric acid-like cycles. He considers these two absolutely separate metabolisms that formed separately. I do not. But that's another matter. And that's going to be explained in the next talk. And I just want to read to you, if I might. All right. I'm sorry. All right. Anyways, the next talk is the formation of the genetic code, the origin of the genetic code in the transition from a thioester world to a phosphate world. That is, I'm going to answer the second big question of the origin of life. That is, this starts off with where did metabolism come from? How did it evolve? How is it related to clays? And how do they couple to each other, clays and the chemistry? Now, the next one is we are now definitely in a thioester world, transitioning to a phosphate world. And in that transition is where the genetic code is evolved. And that's my next talk. So this talk was to set up the origin and evolution of metabolism, the distinction between the thioester world and the phosphate world, and that's it. Is there any questions? Have I answered every one of your hard questions that you, are you convinced that metabolism is an evolved structure? Are you convinced that in some fundamental way that in metabolism as it exists today, you can reconstruct what happened? That is the big lesson. The big lesson is when you went back into the keg and you got rid of phosphate, you ended up with the thioester world, which I include nitrogen. Get rid of nitrogen, you end up in a thioester world pure. And then when you then go back in that thioester world and begin to do much more rigorous studies about boundary conditions, you end up with the reverse citric acid. And I really suggest that you read this book on the transformer. It's a beautifully written book. It's got only one huge error. Everything is done on the proton gradient.
Nick can write.
Pardon?
Nick can write.
Yes, yes.
He's very exceptional.
What?
He's a talented writer.
Oh, he's a brilliant writer.
That's
And-
gonna stick out.
Anyway, I just heard around the grapevine. I don't know whether it's true or not. But there were three people in this autotrophic world. Nick Lane, Martin, and Russell. I think Martin and Russell had a huge fight. Martin now is going through photolysis and photoautotrophy. And Russell says, why have I wasted all these years with you? And Nick Lane, when he acquires his mental agility, will go back to photoautotrophy. if I have a chance, I would just like to read you this paragraph of this last guy. Because if I still can read this, if I have it. Oh, I have it. Well, if I don't have it, I'll send it to you.
That was next time. Okay, you have another lecture.
Well, pardon?
You have another lecture.
Okay, I'll start the next lecture on his opinion of these two worlds. And it's a beautifully written paper, by the way. So, and he really tries to do it in terms of prebiotic chemistry.
Can we let us clap for you?
Okay. Anyways, I hope I've answered any questions that were asked. If not,
We have to leave because otherwise.
I know that. That's why I'm ready to go.
We can continue this conversation at lunch.
Okay, I'll be happy to talk about this. How was it, Ingersoll? Are you happy with this?